Hello, I'm Nino Kioka, one of the co-moderators. It is my great pleasure to start off our WMIF 2021 Disruptive Dozen on cell and gene therapy. We're going to start from number 12. And the number 12 Disruptive Dozen will be Reawakening the X chromosome, a therapeutic strategy for devastating neurodevelopmental disorders. And our panelist is Dr. Jeannie Lee, who's a molecular biologist at MGH and a professor of genetics at Harvard.
Number 12, Reawakening the X chromosome, a therapeutic strategy for devastating neurodevelopmental diseases.
The X chromosome is one of 2 sex-determining chromosomes in humans, and it carries hundreds of disease-causing genes. These diseases often affect males and females differently. That's because males carry a single X chromosome, while females carry 2. Yet in females, one X chromosome is randomly chosen and rendered inactive. Scientists are now exploiting this unusual biology to reawaken the dormant X chromosome, a strategy that could yield much-needed treatments for groups of rare, yet devastating neurodevelopmental disorders, which predominantly affect females.
X inactivation exists to help female cells compensate for the double dose of X-linked genes relative to their male counterparts. While the inactive X is silenced, it is wrapped up and kept in a dormant state. Although X inactivation was once thought to be permanent, scientists are uncovering ways to reverse it. This new approach could hold promise for females with Rett syndrome, a severe X-linked disorder. For the first year or 2 of life, patients appear healthy, but then progressively lose the ability to move and communicate. The genetic culprit? A mutation in the gene MECP2. But only one copy of MECP2 is damaged. The other one is simply inactive. Researchers are now devising a therapeutic approach that molecularly reactivates the healthy MECP2 gene, particularly in the brain.
A similar strategy could also hold promise for other serious X-linked disorders, including Fragile X syndrome and CDKL5 syndrome.
Dr. Lee, what excites you the most about the potential of this technology?
Yeah, so thank you very much. It's so exciting to be here, and I feel really honored to be one of the DD. So we're really excited about this technology for a number of different reasons. So the first is that it's a methodology that really gets at the root cause of Rett syndrome, which is the absence of this critical protein called MECP2. And MECP2, as the video mentioned, is X-linked. So females have 2 copies of it and males have just 1. And that unfortunately means that that males with Rett syndrome do not make it to term most of the time, whereas the girls do make it to term and then have this developmental regression. So it occurred to us a while ago, and others too, that the girls actually carry the cure within their own cells, and that's because they have 2 copies, right? But just one is locked up on the inactive X chromosome. So we reasoned that if there's a way to perturb that inactive X and restore at least part of the expression of MECP2, that we might be able to rescue the Rett syndrome phenotype in these girls. And so another reason we're excited about this is that this is epigenetic therapy, so it's not touching the genes per se, right? So it's not gene editing, it's not gene therapy. We're not introducing any new genetic material into the girls. We're simply coaxing something which is already there to become re-expressed. Now, the treatment is, if it becomes a treatment, would be delivered only maybe 2 or 3 times a year. So it's it would be a very stable drug, but at the same time, it would be reversible. And so if there's at any time a need to, for a time, withdraw the therapy, we could do that and then reapply the therapy down the road. And last but not least, we're really excited about this because all of this is still in the academic lab, but we have some in vivo proof of concept that indicates that at least in an animal model for Rett syndrome that we're able to reactivate MECP2 for a significant phenotypic impact. And so we hope to be able to report more on that in the future. Thank you very much.
Thank you, Dr. Lee. And now for our number 11 technology, disruptive technology. It's entitled Engineering Cancer-Killing Cells that Target Solid Tumors. And our panelist is Dr. Khalid Shah, who Vice Chair of Research in Neurosurgery at the Brigham and Women's Hospital and a professor at Harvard.
Number 11, engineering cancer-killing cells that target solid tumors.
Solid tumors are the most challenging tumors to treat. They grow in a hostile environment that suppresses the immune system, and delivering therapeutics that can reach these tumors is a very difficult task. Now, scientists are devising innovative cell therapies that promise to open new therapeutic opportunities for solid tumors. One approach involves making CAR T cells more like computers, relying on simple logic to decide which cells are cancer and should be destroyed, and which cells are healthy and should be spared. This decision-making, known as logic gating, involves molecular circuits that can identify cancer cells based on the presence or absence of multiple different proteins, instead of relying on readouts of just one protein. By building several logic gates and combining them together, researchers are hoping to pave the way toward targeting new tumor types. Scientists are also devising other groundbreaking forms of cancer-killing cell therapy, including one that uses cancer cells themselves. This approach exploits a remarkable feature. Once disseminated within the body, cancer cells can migrate back to the original tumor. Researchers are now harnessing this rehoming capability, and with the help of gene editing and other molecular engineering technologies, turning tumor cells into potent cancer killers. An early version of this technology uses patients' own cells. Now, the scientists are developing an off-the-shelf version with dual properties, killing cancer cells and modulating the immune system that can be universally applied to patients.
Dr. Shah, what challenges do solid tumors actually bring to the table? And a corollary to that question is, how can the cell therapies you devise help overcome those?
Thank you, Dr. Kyoga. It's a pleasure and honor to be here among you and being one of the disruptive technologies that I want to bring to you. Solid tumors, as we all know, are complex. They have hostile environment, and which is, in majority of the cases, is immunosuppressive. And one of the major problems with solid tumors is also delivery. How do we deliver therapeutics to such tumors? And cell-based therapies offer multiple options. This is one of the upcoming therapeutics that can target multiple components of the tumor, as well as take care of the hostile tumor microenvironment. And for that matter, if you look at the CAR Ts, the recent designer CARs, they target multiple components or multiple antigens, as well as take care of lowering the exhaustion that is caused by the immunosuppressive tumor microenvironment. More recently, we have developed repurposed cancer cells, where we take patient-derived tumor cells and repurpose them and engineer them to express therapeutics that not only kill their own types, but also induce anti-tumor immunity. The ease with which you can isolate tumor cells and perform gene engineering and gene editing on these tumor cells allows you to use them in both autologous and allogenic settings. And in a broad spectrum of things, if you look at the cell therapies, how they can overcome challenges that solid tumors present, you have both autologous and allogenic approaches. And autologous approach helps you in settings where you have limited time between tumor diagnosis and therapeutic intervention. And off-the-shelf therapies are more universal. They can be used in a broad spectrum of tumors. where there is enough time or limited time between diagnosis and treatment. And more importantly, in the delivery settings, cell therapies allow multiple delivery options, and one of the key ones is on-site delivery, where you can combine gene editing, gene engineering of cell therapies with material science, where you can use nanoparticles and biocompatible gels to deliver cells locally. And I think in coming years, what I envision is that we will be using 2 different cell types or multiple cell types for the same tumor, where one will be delivering payload and the other will be targeting the antigens. And this will be actually looking to finally find a cure for solid tumors. Thank you.
Thank you, Dr. Shah. So our number 10 disruptive technology is entitled New Technologies for Delivering Gene Therapies. And our panelist is Dr. Natalie Artzi, who's an assistant professor of medicine at the Brigham and Women's Hospital and Harvard Medical School.
Number 10, new technologies for delivering gene therapies.
A formidable challenge in the field of gene therapy is delivery, getting gene-based therapeutics into the body and into the right target cells. Today, the predominant vehicle for delivering gene therapies is viruses. While viral delivery has some advantages, it isn't ideal. Researchers are now exploring the potential of new delivery methods that could expand the reach of gene therapy. One promising example is microneedles. These micron-scale structures can be grouped together, forming a dense patch. When applied to the skin, the microneedle patch can penetrate the outermost layer with minimal pain and discomfort. This novel delivery method is attracting interest because it can readily access the legion of immune cells that reside in the skin. These immune cells are important targets for vaccines as well as in the treatment of various diseases, including cancer and autoimmune conditions. Another emerging technology involves an implantable device made of biodegradable materials. When placed inside the body, this device can provide localized, sustained release of therapeutics with fewer side effects. The approach is now being tested for the first time in cancer patients using standard chemotherapy drugs that are administered directly at tumor sites, instead of by mouth or the bloodstream. In the future, this method could be customized for the delivery of gene therapy payloads, such as antisense oligos and nanoparticle-based therapies, an advance that could revolutionize cancer treatment, particularly for difficult-to-treat tumors like pancreatic cancer.
Dr. Artzi, where do you think these technologies are going to really have the greatest therapeutic impact?
Thank you, Dr. Kioka, and I'm happy to be here as part of the Disruptive Dozen. So I think that drug delivery systems can be viewed as platform technologies to deliver gene and cell therapies to enhance outcomes in a range of diseases. The goal with these platforms is to deliver the drugs right where they're needed in a desired release kinetics pattern to maximize efficacy while eliminating systemic toxicity. When it comes to microneedles, the most obvious application would be faster administration of vaccines and other therapies in place of intramuscular or subcutaneous delivery. This would enhance the compliance of patients and increase access to the technology in developing countries and in rural locations. We're also looking at ways to use these microneedles to engineer cell therapies, extending their potential beyond molecular therapies. In Biolab, we also devised methods to use these microneedles as a theranostic tool to allow for both minimally invasive delivery of a therapy as well as for sampling of biological fluid for temporal monitoring of molecular and cellular biomarkers in skin interstitial fluid in response to the therapy. So this is powerful because it allows us in real time to monitor the response to our therapy with minimal discomfort to the patient or the mouse in our case. When it comes to drug delivery implants, those systems enable controlled drug delivery pattern and will have the greatest impact in diseases where biological barrier to systemic delivery limit therapeutic efficacy, and in particular, where the target site is accessible or becomes accessible during an intervention, such as in brain cancer or ocular diseases. Local delivery of therapies in this case will increase the armamentarium of therapeutics that are available to treat patients while eliminating systemic toxicity by ensuring an actionable therapeutic window. In my lab, we've already seen in preclinical models how these technologies can revolutionize outcomes in the treatment of the most aggressive and deadly cancers, such as triple-negative breast cancer and brain cancer. We've already seen in the COVID-19 vaccines that the combination of the proper delivery system and the proper drug can change the face of the pandemic. I anticipate the convergence between material science, biology, and medicine will impart the same disruptive outcomes in the treatment and prevention of many other diseases.
Thank you, Dr. Artsy. So our 9th disruptive technology is going to be really welcome news for those of us that like to listen to loud rock music. And it is regrowing cells in the inner ear to treat hearing loss. And our panelist is Dr. Zhangyi Chen, who's an associate scientist at the Eaton Peabody Laboratories at the Mass Eye and Ear, and an associate professor of otolaryngology, head and neck surgery at Harvard Medical School.
Number 9, regrowing cells in the inner ear to treat hearing loss.
Hearing loss is common. In the U.S. alone, some 37 million people suffer from a hearing deficit. Currently, there are no drugs that can halt, prevent, or even reverse hearing loss. Scientists are working on a novel regenerative approach that could restore the cells in the inner ear required for normal hearing, offering hope to millions of patients who grapple with hearing loss. Healthy hearing requires specialized cells in the inner ear called hair cells, which have fine, hair-like projections. These cells perform critical sensory functions, converting mechanical signals from sound into electrical signals that travel to the brain. If the cells are damaged or lost, which often happens with age or after repeated exposure to loud sounds, the body cannot repair them. But researchers have discovered a potential workaround that can stimulate existing cells in the ear to be converted and give rise to new hair cells. This method relies on molecular signals that can reactivate genes that operate during early ear development. When these developmental genes turn on, they turn back the clock, giving adult cells the regenerative capacities of newborn cells. Those powers allow the cells to divide and be coaxed to form hair cells. Scientists are now working to convert this molecular strategy, which is being studied in animal models, into a therapeutic that is safe and effective for hearing loss patients.
Thank you. Dr. Chen, when might a regenerative cell therapy be available for hearing loss patients?
Well, thank you. And first, I would like to, feel honored to be among all those dozen people sitting on the podium. We really take a regenerative approach to derive new hair cells within a year. And actually, in a year, it turned out to be one of the most the toughest organ to regenerate because there's never been any cell division or repair happen. That's why you lose a cell, you lose permanently. So we have actually discovered that by combination of a drug-like molecule, we could really reprogram the cell partially. We don't reprogram to the stem cell stage, but partially to regain for those mature cell regain the property so they can be transdifferentiated into the hair cells. So this has a real great potential in developing a therapy. Our regeneration approach actually is very different from many others. For example, when you're talking about regenerative medicine, normally people can do them ex vivo. You can generate a lot of cells and then transplant in a year. But transplant to a tissue organ. But in a year, that's is unlikely going to work because in a year, extremely intrinsic structure and very small. So all this regeneration had to occur right on site and in precise location. And our approach actually can achieve that purpose. So the other very important aspect is we can regenerate fully differentiate, fully mature in a year, which is truly, for us, is an exciting aspect, because in newborn, even newborn human baby, in a year is fully mature. So for any regenerative method to be applied to human, has to be done in fully mature in a year. So our goal is really within the next few years to really start preclinical study with the methodology we discovered, and depending on the result, we can move towards clinic. So one of the things we can learn from other people, for example, frequency therapeutics is also doing health regeneration, but it's a different method from us. However, there is a roadmap from preclinical to clinical stage, different phases of clinical study. So we can actually learn from them and to speed up the process, as well as avoid some of the pitfall. And we're hoping, in the next few years, we can really see a major progress in making our work towards clinic. Well, we don't really know exactly when that happens, but I can only say with certain confidence, major progress will happen. And one thing I would like to point out is hearing loss is really affecting millions of people, yet there's no single FDA-approved drug. So when you think about the field now, it's almost like 15, 20 years ago. For the blindness, there's no real drugs out there. But look what's these days treating blindness. So treatment for hearing loss, I think, will follow very similar trajectory.
Thank you very much, Dr. Chen. I'm Sue Slagenhop, and I am thrilled to be here as one of the co-chairs of the forum. And our next disruptive technology is CAR T cell therapies take aim at autoimmune diseases. And our panelist is Matthew Frigault, who is Clinical Director of Cellular Immunotherapy at MGH and an Assistant Professor of Medicine at Harvard Medical School.
Number 8, CAR T cell therapies take aim at autoimmune diseases.
CAR T cells have emerged as powerful treatments for some forms of cancer, especially blood cancers. By harnessing the same underlying concept, reprogramming patients' own T cells to endow them with therapeutic properties, scientists are working to develop novel CAR T therapies for a variety of autoimmune diseases. The immune system is designed to protect the body from pathogens and other foreign invaders, but sometimes it mistakenly strikes its own cells and tissues. This autoimmune response forms the basis of many different diseases. Available treatments often involve broadly suppressing the immune system. But this is a blunt-edged approach, which can render patients vulnerable to life-threatening infections. CAR T-cell therapy promises greater precision. Several research teams are engineering CAR T-cells so they can seek out and destroy harmful immune cells, such as those that produce autoantibodies, immune proteins that target and attack the body's own tissues. For example, one team is using CAR T-cells to destroy certain immune cells, called B cells, as a potential treatment for lupus, a serious autoimmune disease that mainly affects women. Another team is testing an experimental CAR T-cell therapy that targets a subset of B cells that make autoantibodies against a critical skin protein. When the immune system attacks this protein, it causes a severe blistering condition. Scientists are also developing CAR T therapies that take aim at other rogue members of the immune system. These efforts could yield novel treatments for diseases with clear autoimmune mechanisms.
So first off, thank you for having me here to discuss this. It's really exciting to see cellular therapies expanding beyond just what we would consider them used for in the oncologic setting. The interesting thing is that when we think about immuno-oncology and immunomodulatory approaches, I think a lot of the limelight's been on cancer. However, when you think about rheumatic disease, rheumatology, and immunology, a lot of the immunomodulatory approaches began in these autoimmune diseases to start with. So now that we're seeing something like CAR T cells, which is currently approved in oncology, moving into the autoimmune space, it's nice to be able to give back, and it's exciting to see it moving into non-malignant indications. So as you saw in the video, in many cases, our approach for autoimmune disease is to just really immunosuppress a patient. That can be through total B-cell depletion with Rituxan, that can be with high-dose steroids, that can be with antibodies against cytokines, but eventually most of the side effects are actually the sequelae of the medications we're giving them themselves. So although we're treating the disease, we're causing other problems in the process. So the hope of using a cellular therapy is that you can more specifically target the autoreactive B cell, the specific antigen. And so we've seen success actually in lupus patients who receive the CD19 CAR T cell. Their numbers actually normalized following treatment. We're seeing early phase 1 data in mucosal pemphigus vulgaris, and there are plans to move that into mucocutaneous disease, to look into things like myasthenia gravis, and also to look at things like antibodies against factor V, which is what you see commonly in hemophilia patients who receive a lot of factor replacement. Now, this opens up the whole world to other autoimmune diseases, assuming we can know the target. It also means that we can potentially move into solid organ donors who have autoantibodies against their potential donor, hemalignancies for bone marrow transplants, and moving it even further, thinking about allogeneic products that are off the shelf and easier to administer. Now, the pie in the sky view is that I'm only talking about B cells here, but there are also subsets of cells called T regulatory cells, and groups like Marcella Moss at Mass General have started to investigate, are there ways to create inhibitory cells called Tregs that can actually shut down the immune response? So it's a normal mechanism that our bodies use to try to deactivate overreactive T cells. And in doing so, can we target tissues and other things that are affected by autoimmune diseases induced by T cells, and can we turn them off to relieve patients of their symptoms without having the broad immunosuppression? So I'm happy to be here and to see things moving into this non-malignant space. I'm really excited to collaborate, as we've seen cellular therapy grow in oncology, with the other subspecialties to get these into more patients, and hopefully pretty quickly.
Thank you, Dr. Frigall. Our number 7 disruption is entitled On the Move, Cell Therapies to Restore Gut Motility. Our panelist is Dr. Alan Goldstein. Alan is the Chief of Pediatric Surgery at Mass General. He's Surgeon-in-Chief at the Mass General Hospital for Children, and he's the Marshall K. Bartlett Professor of Surgery at Harvard Medical School.
Number 7, On the Move, Cell Therapies to Restore Gut Motility.
The human digestive tract, or gut, has its own nervous system. This second brain, known as the enteric nervous system, is comprised of neurons and support cells that carry out critical tasks, like moving food through the gut. When enteric neurons are missing or injured, gut motility can be impaired. Effective treatments are deeply needed. In many cases, surgery is the only option. Now, scientists are developing an innovative cell replacement therapy to treat diseases of gut motility. Donor cells can be isolated from a patient's own gut or from a more readily available source, such as subcutaneous fat, and coaxed to form the progenitors that give rise to enteric neurons. Researchers are also devising off-the-shelf approaches, which could create a supply of donor cells that are shielded from the immune system and can therefore be transplanted universally across different patients. This emerging cell therapy could one day treat patients with gut motility disorders. For example, Hirschsprung's disease is a rare congenital disorder in which infants are born lacking enteric neurons in portions of the gut. The only treatment is surgery. In addition, patients with diabetes often develop problems with gut motility due to declines in enteric neurons. Early research shows that transplanted enteric neurons can also take up residence in the brain. That means these forays in cell therapy for the gut could also help pave a path towards cell therapies for the brain and spinal cord.
Dr. Goldstein, can you share with us your thoughts on how these cell therapies might be delivered? And are there innovations that might broaden their clinical impact beyond the gut?
Thank you very much, and thanks so much for the opportunity and the honor to be a part of this Disruptive Dozen. In terms of cell delivery, we deliver them directly into the wall of the intestinal tract, whether it's the rectum for Hirschsprung disease or the stomach wall for gastroparesis. And currently in large animal models, we use endoscopic ultrasound to accurately deliver it into the muscular layers of the rectum. The very exciting thing that we need to consider here is that we're able to isolate and expand these cells from adult human intestine. That means that each of us has in our gut a large supply of autologous neuronal progenitors. We leverage these currently for neurointestinal disease, which we really have very few treatments for. Our treatments are entirely limited to symptomatic therapies, but none of them actually address the underlying cause of the disease, which typically is either a congenital absence or an acquired loss of neurons in the gut, and that can happen due to infection, inflammation, or neurodegenerative loss, or even trauma. But what's exciting, and I think what the future holds, is recognizing that since we each have an autologous source of readily accessible neuronal progenitors, there's an opportunity here to consider their application for other neuronal diseases in the spinal cord, the brain, and in peripheral nervous system. We've started some studies in peripheral nerve injury to see if these cells can in fact replace injured neurons in the periphery, and we also have done some work with brain injury, whether it's radiation-induced brain injury or traumatic brain injury, and it looks like these cells have the capacity to behave like neurons in other parts of the body, not just the intestine. Thank you again for this great opportunity.
Thank you, Dr. Goldstein. Number 6 is entitled A Flexible, Programmable Approach to Fighting Viruses. Our panelist is Dr. Galit Alter. Galit is a principal investigator at the Reagan Institute and at Mass General and is a professor of medicine at Harvard Medical School.
Number 6, a flexible, programmable approach to fighting viruses.
The COVID-19 pandemic has laid bare the tremendous need for rapidly deployable therapies to counteract emerging viruses. Scientists are now developing a novel form of antiviral therapy that can be programmed to target a range of different viruses, from well-known human pathogens, such as hepatitis C, to those less familiar, such as the novel coronavirus SARS-CoV-2. This new approach to fighting viral infection harnesses a popular family of gene editing tools known as CRISPR-Cas. While CRISPR-based systems have gained attention for their capacity to modify human genes, their original purpose in nature was to defend bacteria from viral infections. As a throwback to these early roots, scientists are now adapting CRISPR tools to tackle a variety of viruses that infect humans. Viral genomes come in different varieties. They can be made of DNA or RNA and can be either single or double-stranded. CRISPR-Cas systems are versatile and can cleave these varied forms. In addition, the tools are programmable, which means they can be designed to target the genetic sequence of any virus. This feature is particularly valuable for addressing the viral variants that can arise, such as those that emerged during the COVID-19 pandemic. Now, researchers are studying the potential of these programmable antiviral agents in the context of several different viruses, including ones that pose significant threats to global health, such as SARS-CoV-2, hepatitis C, and HIV.
Dr. Alter, could you tell us a little bit about which viruses seem most amenable to this approach, and talk about the approximate timeline we're looking at for clinical development?
Thanks, Sue, and thank you to the organizers for the opportunity to be here today. So the list of viruses, I think, is longer than the list of presents that my daughter has on her 10th birthday list. So the opportunities are really immense, and it's really incredible to see this technology really being adapted to trying to deal with pathogens, not just host disease. And the reason is that viruses, by definition, have evolved to evade the immune system. Some have evolved for 2 years, like SARS-CoV-2. Others have evolved for millennia to adapt and evade our immune systems. And so while we think vaccines are the logical solution to trying to eliminate pathogens or alleviate disease and improve health, often vaccines cannot access these viruses in the way that we absolutely need to access them. And so this is where gene editing technology comes in absolutely handy. So when I think about the different classes of viruses, instead of just making a list, I think about those that are intranuclear, so those that are integrated into the genome, where there's an obvious use for CRISPR-Cas to either modify, inactivate, or eliminate the pathogen from the actual host, and those that are actually cause long-lived, lifelong infections outside of the nucleus. And that's how we think about how we prioritize this kind of technology, right? So if we think about diseases like EBV as an example, where we're getting a lot of attention all of a sudden because EBV not only causes lifelong infection that we thought previously was innocuous, we now know that it causes NPC, it causes liquid tumors, it causes many tumors across the entire globe, but also now has been implicated in causing MS. So thinking about ways that we can interfere with this virus for which vaccines have been ineffective. We've tried for 30 years to design vaccines that could prevent infection with Epstein-Barr virus, which has not been accessible and have not been successful. But using CRISPR-Cas, now interfering with the actual genomic persistence of this particular virus, has really changed the game and changed the balance on our side to be able to eliminate this virus once it has integrated into the host, both in people who are potentially more susceptible to developing the disease within familial groups of individuals that are more prone to developing, let's say, NPC or nasopharyngeal carcinoma, as opposed to those who may not have that predisposition. And so that's one example of how we've been thinking about this. But this applies to hepatitis B virus, it applies to CMV, it applies to HPV papillomavirus, for which we're looking endlessly for ways that we can actually excise these viruses or inactivate them within the human host. The other class of viruses that we think about when we're prioritizing how to use this new CRISPR-Cas technology, because it's absolutely flexible and so easily adaptable, are the viruses that do insert themselves into our genomes. And so they're, my obviously favorite for life is HIV, for which 40 years of research and endless numbers of clinical trials continue to fail to induce protective vaccine-induced immune responses. And we've come to realize that development of these gene therapy editing technologies that can inactivate the virus once it has integrated into our genome is absolutely critical as an intervention to end the pandemic caused by HIV. With a 95% mortality rate with HIV infection, it is one of the most deadly pathogens that we have on this planet. And so thinking about ways that we can excise segments, the LTR alone, just this tiny little segment, is enough to completely inactivate the virus and really stop it on its track. The challenge there, of course, is just how do you get that CRISPR into the right cells? And so this is where we're working endlessly to think about how we can target cells with this CRISPR-Cas technology to access that virus of great interest. But the last category I want to leave you with is this idea that there are also a completely different class of viruses that we think about when it comes to thinking about improving human health. And these are not classical pathogens. But we think about how we can use CRISPR-Cas technology, again, to modulate the immune system, remodel the RNAome or the host expressome, let's say, and that is through the engineering of endogenous retroviruses. 50% of our genomes is formed of these types of elements that are archived viral DNA that sits within us and modulates the way that we express proteins, the way that we encode RNAs, how we express them. And so thinking now about how we can begin to target those elements, those viral elements that live within our genome, gives us an endless opportunity to think about how we can intervene, remodel, and improve outcomes across a much vast or larger array of different potential infections and disease. So the list is long, but I hope that gives you an idea of really where we're thinking this technology can take us into thinking about direct pathogen interventions, but also a much broader landscape of diseases that we feel like we can interfere with.
Thank you, Dr. Alter. Our number 5 is realizing the promise of gene therapy for brain disorders. And our panelist is Feng Bei, principal investigator in the Department of Neurosurgery at the Brigham and assistant professor of neurosurgery at Harvard Medical School.
Number 5, realizing the promise of gene therapy for brain disorders.
Gene therapy holds enormous promise for serious and currently untreatable diseases, including those of the brain and spinal cord. A commonly used vehicle for gene therapy, a virus called AAV, cannot readily access the brain when injected into the bloodstream. That's because it cannot penetrate a major biological roadblock, the blood-brain barrier.
While there are alternative routes to the brain, they are invasive, costly, and not feasible for reaching deep regions.
Now, researchers are engineering new versions of AAV that can cross the blood-brain barrier. Using various molecular strategies, a handful of teams have modified the protein shell that surrounds the virus so it can gain entry and become broadly distributed within the brain. These modified viral vectors are now under development and could begin clinical testing within a few years. Scientists are also tinkering with the inner machinery of AAV to sidestep potential toxicities. For example, a specialized group of neurons called the dorsal root ganglia can degenerate following treatment with AAV-based gene therapy in animal models. To minimize the possible risks to humans, researchers are now working to tweak the viruses so they are less active in these neurons. These molecular feats represent a significant step forward for the gene therapy field. With a safe, effective method for accessing the brain, researchers will be able to devise gene therapies for a range of neurological conditions, including neurodegenerative diseases, cancers, and devastating rare diseases that lack any treatment.
Dr. Bay, based on your work, which disease or disease areas do you think these technologies will be applied to first?
Thank you for the questions. First of all, I would like to emphasize that the progress described in the video actually made by the whole field, so we are still learning to be a player. Right now, available data on this new blood-brain barrier penetrating AAVs seems to suggest two take-home messages. First, these new versions of AAVs are approximately 5 times more efficient than the gold standard, quote, and AAV9 in accessing the brain in a non-human primate, which is significant. Second, these new versions of AAVs, however, are still not efficient enough to target all the cell types or regions in the CNS. So given these results, one may be interested in first explore 2 disease areas or diseases. A is CNS diseases that the cells are able to cross-correct each other. For example, for some form of a lysosome storage diseases, since one, in theory, only needed to collect a fraction of brain cells in order to provide patient benefit. B would be CNS diseases that can that predominantly affecting the cell types or regions that these AAVs have a reasonably high targeting efficiency. So in addition to the motor neurons, which are known to be a convenient target for AAV9, now the Purkinje cells in the cerebellum may be the next to target.
Great. Thank you very much, Feng Feng. So I have the distinct pleasure of introducing the last 4. I promise you it's not a baker's dozen, so there's only 4 left. And that is entitled Harnessing, the next, number 4, Harnessing the Power of RNA to Treat Brain Cancer, and that will be presented by Anna Gruchevsky, who's an associate professor at the Brigham. Anna.
Number 4, harnessing the power of RNA to treat brain cancer.
RNA, the close chemical cousin of DNA, is widely known for its helper functions, carrying messages from one part of a cell to another to make proteins. But scientists now recognize that RNA plays a more central role in biology and are tapping its hidden potential to create potent new therapies for a range of diseases, including a devastating form of brain cancer called glioblastoma. This cancer is extremely challenging to treat. It lies within the brain, which is difficult to access with drugs and other therapies. And it has proven highly adaptable, overcoming practically every treatment that's so far been deployed against it. But new approaches that either target RNA or mimic its activity could hold promise. For example, researchers are investigating an intriguing class of RNA molecules called microRNAs. One team identified a trio of microRNAs that play important roles in healthy neurons, but disappear when brain cancer develops. These microRNAs can be stitched together into a single unit and delivered into the brain using a virus. Initial studies in mice reveal that this therapeutic can render tumors more vulnerable to existing treatments, including chemotherapy. Another team is also exploring microRNA-based therapy for glioblastoma. The researchers discovered that glioblastoma cells are addicted to a particular microRNA called miR10B. Blocking its activity causes the tumor cells to die.
Now, scientists are working to develop a targeted therapeutic against miR10B that can be tested in clinical trials.
Great. So these are one of the areas that has promoted innovation just because of their challenge. And so, Anna, the question goes is, what excites you most about the potential of RNA therapeutics?
Thank you, Ravi. Thank you. I'm really happy to be part of this panel as well. So we are very excited about RNA, potential of RNA therapies, and we have talked quite a bit during this 3 days meeting about RNA therapeutics, and you have heard amazing speakers, and there were really great panels about RNA therapy, so I hope some of you may share our excitement now. Again, you know, I started with this message on Monday morning, and I want to reiterate it again. I think it's a super powerful idea. 2% of our genome encode for proteins, and that entire biomedical research has been focusing on this 2% of human genome until very recently, with very few exceptions. And now we have this full realization that the majority of our genome, human genome, is transcribed to various classes of RNAs, and this provides us with this tremendous potential for identifying new targets. There are these very powerful coding and non-coding regulatory RNAs in our genome that could be targeted, and the technology is available, at least partly. Of course, there is a long way to go, but we know the rules, a lot of rules of this targeting, and the technology, as we know now, based on the COVID vaccines, is scalable. So it's extremely exciting. I want to mention that RNA therapeutics are different from protein-based therapeutics. Those are informational drugs. It means that basically the information needed to design a drug is encoded in the RNA sequence itself. And what it gives us is actually that we can now target what we could not target before. Proteins that could not be targeted before now can be targeted via their mRNA targeting. And beyond the mRNA, we can target lots of other RNAs using several strategies that, again, will be based on the RNA sequencing. And it will be probably fairly fast and rapid process comparing to the designing of protein targeting strategies, recombinant proteins, antibodies, and so on. And another thing, the strategies are also quite flexible and adaptable to new variants. So just as we learned about the possibility of designing new vaccines for new COVID variants, similarly, we could think about adapting and with small changes, making a smart design for new variants emerging with new diseases. So our work is really on brain tumors, and we believe that we have really good RNA targets, non-coding RNAs, as therapeutic targets that would help us to treat really incurable diseases such as glioblastoma. But more broadly, RNA therapeutics are here and hopefully will be developed and will be really will be the major class of new therapeutics in the near future. Thank you.
Great. Thank you, Anna. And let me just say thank you for sharing your science and your excitement in this area with us throughout the meeting. So number 3 is entitled Cell Therapies to Conquer Common Forms of Blindness, and that'll be presented by Michael Gilmore, the Chief Scientific Officer at the Mass Eye and Ear.
Number 3, cell therapies to conquer common forms of blindness.
The eye has been a proving ground for pioneering gene therapies. It is also fueling new cell-based therapies that can restore sight to blind patients. Rather than fixing the genetic causes of a particular disease, These cell therapies offer a functional cure by replacing critical cells that have been lost or injured. One approach involves stem cells from the retina, the specialized tissue that lines the back of the eye. These stem cells can give rise to light-sensitive cells, called photoreceptors, which are required for healthy vision. But the stem cells cannot generate cell types that are not normally found in the retina, like bone or muscle. Scientists are now harnessing retinal stem cells to develop treatments for incurable eye diseases, including retinitis pigmentosa. Because the immune system doesn't patrol the eye as aggressively as other parts of the body, retinal stem cells from unrelated healthy donors can be transplanted into patients with vision disorders. Researchers are also developing cell therapies that harness patients' own cells. With cell reprogramming technologies, patient-derived cells, for example, from blood or skin, can be converted into almost any cell type in the body, including retinal cells. In addition, scientists are now testing a novel treatment that utilizes stem cells from the cornea, the clear covering at the front of the eye. Corneal stem cells from a patient's healthy eye are removed to repair the affected cornea of the other eye. The potential benefits are significant, as corneal damage is a major cause of blindness across the globe.
So we certainly heard some of the answers to this question already in the video, but Michael, help us put a punctuation mark on this point. And what are some of the features that make the eye unique to gene and cell therapies?
Great, Robbie. Exactly. We believe the eye is ideal for a number of reasons that were highlighted and some that I'll add to. First of all, it's a surface accessible complex organ. And that means that you can directly access it, and it's transparent. And so you can observe intraoperatively the placement of cells, make sure they're placed in the right position. You can non-invasively follow that up the next day to see whether they're engrafting. You can continuously follow it without disruption. A great virtue of the eye is that it actually is a projection of brain tissue out to the surface, but it's one that we can approach locally. So we can put a lot of cells or a lot of drug on target with very little systemic complication. The last virtue was touched on also. It's like the brain, there's a blood-retinal barrier that limits the influx and efflux of cells or trafficking into and out of the eye. So by and large, they stay where they're placed. It also is immune privilege, which opens the door to allogeneic therapies, which affects manufacturing processes. Finally, I would say that the treatment, when successful, has a tremendous impact on a person's life in terms of life quality. So it's a great first in site for therapies and new technologies. The impacts are great, and so that's the ideal. That's why we think it's ideal.
Thank you, Michael, and thank you for your leadership at the Eye and Ear. Okay, so number 2 is entitled A Gene Editing Solution to Increase the Supply of Donor Organs, and that'll be presented by Jim Markman, the head of transplant surgery at the Mass General.
Number 2, a gene editing solution to increase the supply of donor organs.
In the U.S. alone, more than 100,000 people await a life-saving organ transplant. But an inadequate supply of donor organs leads to prolonged waiting times, and many patients die before receiving a life-saving organ. The only foreseeable solution to this crisis is xenotransplantation, harvesting organs from animals and placing them into human patients. The idea has been around for centuries, but xenotransplantation has only recently become feasible due to advances in gene editing technology, which makes it possible to remove, insert, or replace genes with relative ease and precision. This molecular engineering is necessary to sidestep the human immune system, which is highly adept at recognizing foreign tissues
whether they are from a distantly related animal, such as a pig, or even a family member. Over the last 20 years, scientists have been working to devise successful gene editing strategies that will render pig organs compatible with humans. They have removed pig genes that inflame our own immune system and inserted human genes that help suppress it. Researchers have also ridded pigs of all genes associated with a type of virus, called PERV, which stands for porcine endogenous retrovirus. These viruses can infect human cells, so removing all traces of them may be critical for enabling safe and effective xenotransplants. Over the last year, the field has taken another major step forward, transplanting gene-edited pig organs, including the heart and kidney, into humans. While extensive clinical testing is needed before xenotransplantation becomes a reality, that future now seems within reach.
Great. So Jim, the question to you is, what are some of the challenges that still need to be overcome? And you heard that obviously from the video as well. So please, Jim.
Thank you, Robbie. It's a great honor to be on the panel here today and to talk about xenotransplant. The recent pig heart transplant was really a momentous event for the field, the first of its kind, and I think has really stimulated a lot of interest. I think there's three key points that need to be addressed for the field to continue to move forward. First is identifying the correct edits to make pig organs compatible and overcome the xenobarrier. And the xenobarrier is this combination of antibody-mediated targeting of the endothelium of the pig, plus incompatibilities in coagulation and complement between the species. And this has been the focus of the different edits. And I think this is the area in which the field has made the most progress. And now it's quite common for a pig organ, a heart or a kidney, to survive for years with good function in a large animal. So there's been really great progress there. The second issue that may be more challenging is the safety issue. As was mentioned, the PERV problem has been taken care of by CRISPR editing. And at least in one pig line, there were 63 copies of the PERV gene, and all 63 were disrupted in a single treatment of the cell. So the PERV problem has been taken care of. There are other potential pathogens. In the recent pig heart transplant, about a month or so after the transplant, the patient was noted to have a porcine CMV infection, so cytomegalovirus. And this was supposed to have been a clean animal that had been tested and was seronegative for all these. So I think disease transmission still remains a barrier that we'll have to do better on. And then finally, the immunosuppression is key. In all of the pig large animal studies that have been successful, immunosuppression to target either CD40 or CD40 ligand was used. There are anti-CD40 and CD40 ligand agents that are going through clinical trials, but they're not yet approved. So until those agents are available, those agents need to be available, I think, for xenotransplantation to succeed. But I think it's inevitable that it will, and it'll be a great help to all our patients.
I'm going to use the prerogative to ask you one more question, Jim. We've seen heart. What do you think is going to be the next organ in this space that we're going to?
I think kidney is very close. Yeah.
I'm a nephrologist, so I was hoping you were going to say that. So that's great. Good. Thank you, Jim. Okay, well, last but not least, restoring sight by mending broken genes. This is going to be presented by Dimitrios Vavas, who's from the Mass Eye and Ear, professor of ophthalmology at Harvard Medical School.
Number 1, restoring sight by mending broken genes.
Roughly 200 genes are directly linked to vision disorders. In the last several years, groundbreaking new gene therapies have emerged that can compensate for faulty genes in the eye by adding new, healthy copies. A molecular fix that promises to restore sight to those who have lost it. As remarkable as these new treatments are, they have important limits. For example, some genes cannot be replaced because they are too large to fit in the delivery vehicle, a virus known as AAV, which carries them to cells within the eye. Now, researchers are testing an experimental therapy that could help overcome this hurdle. Instead of replacing defective genes, it repairs them, like a word processor at its misspelled words. The approach, known as CRISPR-Cas9 gene editing, could open the door to treating genetic forms of vision loss
that are not suited to conventional gene therapy, and a host of other medical conditions. A clinical trial is now underway to evaluate a CRISPR-Cas9 gene editing therapy for a severe form of childhood blindness known as Leber congenital amaurosis type 10. The condition is caused by mutations in a large gene called CEP290. Currently, there are no treatments. Although this treatment is still experimental, it's already historic. It is the first medicine based on CRISPR-Cas9 to be delivered in vivo, or inside a patient's body. Similar gene editing therapies are also under development that correct genes within blood cells, which are first isolated from patients, treated with the gene editing medicine, and then infused into the body.
So Dimitrios, unfortunately, Michael took all of your talking points, but I'm going to ask you this question, which is, what fraction of patients are going to benefit from this type of approach?
Yes, thank you very much. It's a big honor to be here as a panelist. And I thank Mike for the introduction, why the eye is so good. I also feel privileged and honored to be part of the Mass General family, but also part of the ophthalmology family, because ophthalmology has the first in-human successful gene therapy in Leber's congenital amaurosis type 2. And now ophthalmology is leading the way in the first human application of CRISPR-Cas9. The video touched upon that this technology can apply to patients that have large gene defects in genes that are large, that they cannot fit in an AAV virus. But also it can help patients that they have what is called autosomal dominant disease. Autosomal dominant disease is a disease that only one copy of the gene can be faulty and creates a protein that is dominant in its negative effects. And the current gene therapy technology cannot apply to these patients. So gene editing, and especially prime editing, can apply to a lot of these patients. Now, in theory, this technology is suitable for everybody, but in reality, it's different because we do not know the gene defects for about a quarter of the patients with retinitis pigmentosa. And then even for the patients that we know the gene defect, there has to be certain requirements about the sequence of the genes next to the mutation. This technology is not efficacious to all gene sequences. So the efficacy of this therapy still needs to be worked out, as well as the off-target effects of this therapy.
Great, thank you very much, Dimitrios. So let me ask the audience for a round of applause for these 12 incredible presentations. So as much as we hate to say this, we have come to an end, so just a few closing remarks. First, as you saw, we started this conference on Monday with science, and we're ending with science. And that's where really this area begins. It doesn't end there. It actually then is stimulating the audience and stimulating the partners that we've had as part of this particular conference. And thanks to Chris and his team, and that's what all the conferences at the World Forum have always done. We've started and ended with science, but that doesn't take away from the fact that we've had some incredible partners in this. One, Bank of America, as Chris mentioned. We've had over 1,000 registrants for this particular conference from, I'm told here, 26 different states, 12 countries, 350 different organizations. Absolutely incredible. We talked about science, but again, in between, we talked about regulatory, we talked about industry, we talked about biotech, manufacturing, and we talked about the capital markets together with our partners from Bank of America. So thanks to all of you. Thanks to the 150 speakers. Thanks for staying till the end. I'm going to hand it to Sue to make some other closing remarks.
Thanks, Ravi. On Monday morning, we began by hearing from Mass General Brigham investigators on their compelling gene and cell therapy research that could significantly impact the field in future years. The new Doctor is In sessions provided a deeper dive into the scientific challenges and opportunities in gene and cell therapy. Each session was led by a Mass General Brigham clinician and a Bank of America analyst, bringing new perspectives on how clinicians and industry can work more closely together to bring new and innovative developments to the field. We heard from healthcare, investment, and government leaders about the regulatory and manufacturing challenges, the need to improve data sharing across healthcare systems, the need to lower costs, and the need to increase accessibility. As a scientist who focuses my work on ultra-rare disease, I believe that the gene therapies have remarkable power, but admit to being frustrated at times by the difficulties in moving things forward for ultra-rare disease. So I would just like us all to leave to thinking about the fact that we're going to have to work together, both in academia as well as industry, to make sure that as we advance these technologies, we figure out a way to make sure that everyone benefits, no matter how rare their disease. And thank you all for being here, and I'll turn it over to Nino.
So I have the pleasure of making the final remarks, and it's great to be here towards the end of this pandemic, we hope. In fact, we've had some really valuable conversations about COVID. We've discussed how we shift our mindset from eliminating it to actually living with the virus, how important it is that we globally distribute this vaccine, and what is the role of RNA and RNA therapeutics in this as well. But why discuss COVID in the middle of a gene therapy conference? Well, think about it. Until very recently, gene therapy was a therapy that we were discussing for very rare monogenic diseases, and that's what it has been FDA approved for until COVID. Now, each one of you has been exposed and has tested gene therapy, either whether you had the RNA vaccine from Pfizer or Moderna or the Johnson & Johnson adenoviral vaccine. We all are now have had multiple doses of gene therapy. So gene therapy is now a global therapy. And I really think that this means that the opportunities for investment in this field are really becoming more enriched. Here at Mass General Brigham, our new partnership with Bank of America really is going to allow us to really expand the programming and emphasizes our role in New England as a powerhouse in biopharmaceutical development. This could not really happen without our strong culture of working together, of collaboration, and we look forward to continue to build a symbiotic relationship with Bank of America as well. All of the content from the past 3 weeks is available to you on YouTube. We encourage you to watch it and continue these conversations. Next year, we will be at our new location, the Westin Boston Seaport District, on June 12th and 14th. And if you actually register this week, you will get a discount on your conference rate. And finally, I was going to ask you as the audience to please stand up and let's have a round of applause for Chris Coburn and the steering committee that really put together this great and rich program. Thank you, Chris. Thank you, steering committee. To next year.